transfer approach to estimating ET. So what I've shown here is the cross-section of a leaf, and in the bottom are a couple stomatal openings here between cavities within the leaf and the atmosphere. So that's the atmosphere out here. And the important point is that these mesophyll cells surrounding all the cavities in the plant are saturated with water. All of these cells are saturated with water, and that drives a constant evaporation off of their surfaces into these cavities in the plant. So all of these cavities in the plant are constantly being fed by water evaporating off these saturated cells to the point where all of these cavities, they're very small, are almost always, or essentially always, saturated with water vapor, which I'm indicating with this sort of blue color here. So all of these cavities in the plant are saturated with water vapor. And assuming that the atmosphere outside the plant here, outside the stomatal openings out here, assuming that that atmosphere is not saturated with water vapor, that will set up a concentration gradient from a point inside the plant here through the stomata to a point out here in the atmosphere. There will be a concentration gradient that will drive the diffusion of water vapor through the stomata into the atmosphere, and that's essentially evapotranspiration. And according to if we're assuming that the control, the rate at which evapotranspiration can happen, is determined by that rate of mass transfer, we can therefore describe evapotranspiration as simply being governed by the diffusion equation, which you might remember is J here is the rate of mass transfer. That's just the rate of evaporation through the stomata, which is equal to some diffusivity. I'm just going to call that C. That's the diffusivity. I'm going to call it C star here for a second, times the gradient in the concentration. And in this case, I'm going to measure the concentration here in the middle of the cell, in the middle of the little cavity inside, and measure it outside in terms of the water vapor pressure E. E is water vapor pressure. Remember, that's one of the ways we can describe the concentration of water dissolved in the atmosphere, is water vapor pressure. And that's typically how it's done. So this diffusion type equation looks like the constant times the gradient in the water vapor pressure, and that is just the water vapor pressure difference, the water pressure on the inside here, minus the water vapor pressure on the outside, divided by the distance apart. And that's just this distance here. I'll call it delta x is just this distance here. And that's not really important. It's some small distance that separates the interior cavity with the atmosphere through the stomata. Remember, however, that inside the little cavities in the plant, the water vapor is always at saturation, right? This is always equal to the value of the water vapor pressure at saturation. So we can actually write this as the diffusivity constant times, I'm just going to lump the delta x into this too, times the difference between the saturated water vapor pressure and the water vapor pressure outside the stomata in the atmosphere. That then is the form of the mass balance approach estimate of evapotranspiration. Now, so how fast can this process happen? Well, it's going to depend on two different things, I hope you can see. First of all, I hope you can see that the rate of this process is going to be limited by this difference here. We call that the water vapor pressure deficit. And remember that if that deficit were, for example, to be zero, meaning that the water outside is the same as the water inside, then there's going to be no movement of water vapor by diffusion. And ultimately, that's what's going to happen, because as this water vapor moves to the stomata, it accumulates out here right outside the stomata, and at that point, right, we've stopped the process, because now there isn't any longer any concentration gradient between the interior and the outside of the stomata. And so for this process of continued mass movement to occur, we've got to get rid of this excess saturated air. So we have to get rid of that air somehow. Now, why would that air move? Well, again, it's accumulating out here, but there will be diffusion, right? It's going to diffuse from high concentration to low concentration, so it's going to slowly move away by the process of diffusion, but that could presumably take a fair amount of time. So that's one factor that's going to allow it to move out of the way. But another one, and a really important one, is that if there is wind, and especially if there's a variation in wind, which produces these kind of swirling eddies of wind, the wind will sweep this saturated air away from the outside of the stomata, clear out all that air, and we can, whoops, let me get there, let's see what I'm trying to do here, clear out all that air, and we can, try again here, yeah, clear out all that air, and once again permit diffusion to occur, allowing evapotranspiration to continue. So the two factors that are going to determine how fast evapotranspiration can occur are, one, whether there is a water vapor pressure deficit and how large it is. The larger the water vapor pressure deficit, the stronger the concentration gradient, and we know from Fick's law of diffusion that means the faster the rate of diffusion through the stomata, right? So the bigger the gradient, the faster water is diffusing through this stomata. The other factor is how quickly can we get rid of that saturated air on the outside of the plant. The stronger the wind, the stronger the concentration gradient, the more quickly we will sweep that saturated air away and permit the process to continue. Now we can look at that water vapor pressure gradient on the same diagram we've used here in the past to look at the saturation curve for water vapor as a function of temperature. Here's a graph you've seen before. I'm graphing the water vapor pressure on the y-axis in pascals. The blue line represents the level of saturation, the saturation water vapor pressure as a function of temperature. So for example, let's say we had an air mass, an atmosphere that had a temperature of 20 degrees, and it had a water vapor partial pressure of 1,000 pascals. It would plot right here. So this would be the vapor pressure outside in the atmosphere. Inside the cavities in the plant, we know that they're always at saturation, so we can draw that right up here. This then is the value of the water vapor inside the plant. And so the water vapor deficit that drives evapotranspiration, it's part of our equation, is just simply this vertical difference here. That's the water vapor pressure deficit that drives evapotranspiration and causing water to diffuse from the inside of the plant to the outside. Notice also that this depends on temperature. If we took the same air mass and we cooled it down, so it was, say, later in the day when it got colder, and it cooled down to, let's say, 12 degrees C here, we would have a much smaller water vapor pressure gradient, right? Once again, the interior of the plant's at saturation. It's always along that line. And now at 12 degrees, we'd have a much smaller water vapor pressure deficit. Is that what I said? I think I said gradient. I meant deficit. The water vapor pressure deficit. So it depends on temperature, which is why it should not be surprising then that this whole equation is very strongly depends on the temperature, because the temperature determines the value of the saturation water vapor pressure, and that is a big determinant of how quickly water vapor diffuses from inside to outside of the stomata. Now at this point I want to make a couple quick comments about what do we mean by this diffusivity term, C star. That describes, remember, the ease with which water vapor can diffuse through the stomata. And so it's actually going to depend on several things that are very specific to the plant. It will depend on the size, for example, of the stomata. Bigger stomata will have a less resistance to movement. So smaller stomata in certain kinds of plants will offer more resistance. It will depend on the density of the stomata. If a plant has a lot of stomata per leaf versus few stomata per leaf, that will change the ability of water vapor to move through the stomata, or the amount of water vapor that can move through the stomata given a given water vapor pressure deficit. It will, let me just density of stomata here. This also depends simply on the kind of plant, right? That's kind of plant determines the size and the density of the stomata. Another factor that is related, actually the big factor here, these are all relating to the plants itself, and I should add the size of the plant here Grass, for example, will offer a very different type of resistance or conductivity than an oak tree. Not only does an oak tree have many more leaves per area, but the oak tree is much taller. And the higher the tree, the more you're going to see differences in the wind velocity from at the top of the tree to the bottom of the tree to the top of the plant to the bottom of the plant. And it's those variations in the velocity of wind that actually drive this turbulence that causes the wind to aid in the removal of the saturated air. So the big factor here, the really big factor here, is wind speed. And actually, I'm going to underline that, and also differential wind speed. What I mean by differential is the difference in wind speed between the top and bottom of the plant, because that is actually what causes the turbulent eddies that aid in moving away the saturated air. So this is really the big factor that gets incorporated into this diffusivity constant here that that very strongly affects the rate at which water vapor can be removed from the plant into the atmosphere. And that's it. That's the essential features, I should say, of the mass balance approach to estimating evapotranspiration.